The virus that causes COVID-19 spreads person to person mainly through respiratory droplets, from coughing, sneezing, or talking. These droplets have the potential to spread and infect another person who is within 6 feet. Once the virus enters the body, it will cause infections in the respiratory mucosa, then spread to the host alveoli cells. In healthy individuals, angiotensin-converting enzyme 2, or ACE2, will break down the protein called angiotensin 2, but when the virus binds to ACE2, it will disrupt the enzyme's ability to break down angiotensin 2. And so, the accumulation of the protein angiotensin 2 will increase inflammation, leading to alveoli cells' death, thus reducing the individual's oxygen intake. Let's take a deeper look at the entry and replication processes that the virus undergoes. As mentioned before, the entry is driven by interactions between the spike protein and the host cell's ACE2 receptor, similar to a lock and key mechanism. With the help of cellular proteases like this pink one, a proteolytic cleavage event will aid in the fusion of the virus and the cell. There are actually 2 ways the virus can enter, either through endocytosis like this or another mechanism called fusion. Once in the cell, the genetic material of the virus, which is encoded as a 30 kilobase single-stranded plus sense RNA, will be uncoded and released into the host cell. This plus sense RNA will directly function as an mRNA that will be translated by the host ribosomes. Here is a closer look at the different sections of the messenger RNA before translation. Open reading frames in the mRNA will be translated by the host ribosome into large polyproteins, 1A or 1AB. Then, these polyproteins will be proteolyzed to produce up to 16 smaller non-structural proteins. You may be wondering, what exactly is cutting these polyproteins? They are being cut by 2 proteases called papain-like protease and main protease. It is important to note that these proteases themselves are non-structural proteins, specifically NSP3 and NSP5. However, unlike the other non-structural proteins, these proteases are able to cleave themselves off the polyprotein without any help. After they are lysed, they will act like scissors to cut the other non-structural proteins from the polyprotein. One of these newly cut non-structural proteins is NSP12, which will act as an RNA-dependent RNA polymerase and will attach to the plus sense RNA and undergo replication to create a minus sense RNA, also known as an antisense RNA. This will lead to 2 pathways involving the antisense strand. The first pathway creates the genome of the viral offspring. To do this, the antisense strand will be replicated back into the plus sense RNA. The second pathway uses discontinuous transcription, in which multiple subgenomic RNAs are transcribed again by the RNA-dependent RNA polymerase. The subgenomic RNAs then enter the endoplasmic reticulum to be further translated into structural or accessory viral proteins, such as the spike, membrane, or envelope proteins. After the viral proteins are made, the offspring's genome and nucleocapsid proteins in the Golgi complex will meet with other structural proteins to form small vessels to be exported out of the cell through the process of exocytosis. This is the basic structure and overall replication cycle of SARS-CoV-2.